Hi everyone, welcome back to Editing Humanity. My name's Connor. I'm a biomed student here at NAU, and today I am joined with Dr. Hodge, who was my professor for Bio 375 infectious diseases, one of the most interesting classes I've taken. And yeah, how are you, Professor Hodge?
I'm fine. How are you doing? Good to see you again.
Pretty good. Good to see you too.
Yeah.
Um,
So I guess you wanted me to give a little brief intro, and yeah,
yeah.
okay. Um, I'm Dave Hodge. I'm an adjunct professor at NAU. Before I came out here, I'm retired. You can tell by looking at me, I guess. I'm retired now. I retired from federal government after 35 years, but about 20 of those years were spent as a senior program manager at the Department of Homeland Security in what we call Chem Bio Division, which is part of Science and Technology Directorate. And while I was there, we set up an operation to do forensic analysis of potential biological select agents and biological terrorism incidents. And that was at Fort Detrick, Maryland. And then at the conclusion of the anthrax litter case, which, by the way, was solved up here by Paul Kimes' group at PMI over and back at a Target store there. That is probably one of the reasons I came out here because I like Flagstaff so much. But after that, I worked for, I guess, 12 years doing the biosurveillance work for a couple of different federal agencies, including the United States Secret Service, who has obviously an interest in biosurveillance and biodefense. Uh, I'm a graduate of the University of Maryland for my undergraduate degree, and I went to Johns Hopkins University in Baltimore, a small private school in Baltimore, and that's where I got my PhD from. And my PhD was actually in sort of like biochemistry and virology, uh, molecular virology, we called it. And a lot of my work was on uh, control of virus expression and kind of classic virology, but also at that time some of the newer biological methods. So I ended up doing a lot of work at the National Cancer Institute for 17 years, and then another, I guess, 20 years on top of that. DHS. We actually set up part of DHS when I was there, and so after 36, 37 years of that nonsense, I was ready to retire. But that's how I ended up out here. And they wanted me to teach a couple of advanced courses, which Connor, you took one. And the other course I teach, I can do a plug, right? The other
Yeah, sure.
course I teach is clinical microbiology, which is kind of like, all the kind of medical issues people would run into. A lot of kids go to medical school. They don't really train you up on that. They
Yeah.
expect you to pick it up as you go along. So I just kind of made up a course out of my head and just trying to stay away from the kind of molecular biology and general micro stuff. And just like, this is what the infection will look like. This is a syndrome. This is how you identify it. This is how you treat it. And, this is how you interpret all these results. So it's been well received. It fills up pretty quick. And
Yes.
there's always going to be one person in the class who's saying all he does is talk about himself. Well, I mean, I've been looking at this stuff for 45 years. So, I know a little bit more about it than some people. So, it's, yeah, it keeps me busy. Otherwise I'd just be sitting down here watching TV or something during the day. Yeah. So Connor, you had a bunch of questions for me. I noticed, and I was just going to kind of pull this screen down a little bit. I'll let you go through the list and ask me what you want to ask me. Okay?
Yeah, of course. Yeah,
Yeah.
so I guess my
Okay.
first question and foremost is, CRISPR is pretty controversial as of late,
Yeah.
just
Yeah.
based on like just all the stuff it can do. What are your personal thoughts on CRISPR as someone who has like worked for the Department of Homeland Security and the government a long time?
Yeah. Well, I started out, like I said, in just graduate school, and I've actually made recombinant viruses. That was one of my fortes when I worked at NIH. Uh, You know, viruses are like the biggest non-union workforce you can get, and we didn't have CRISPR when all this started out. We just did use something called homologous recombination, where we could just take a gene that we wanted to insert or knock out in a virus or a cell or something, and we could make a plasmid with that. And then you would take maybe like 1,000 base pairs on either side of where you wanted to insert that in the viral genome, and then you would just co-transfect that into a cell, and they would recombine, and you could screen for the recombinants, and then you had a virus that was a recombinant virus. So we could insert genes all the time. We could also make mutations. That actually is very easy. That's how we started out, just doing like just combination, and then we started doing PCR. A lot of viruses are small enough in their genomes where you can just make PCR primers and go into the genome and just put one plus and minus strand, and then just do PCR all the way around with a long processive enzyme and make a new virus. So I mean, this is how you, if you were going to do it, this is how you could have made the COVID-19 virus. Insert a furin site in there, which makes it much more efficient to infect. And which some people, not me, some people would say that's probably how they did it.
Yeah.
So the CRISPR, that's in biology we have like white hat biologists and black hat biologists. There's always some idiot out there that's going to do something stupid with a virus. I mean, I direct your attention to many science fiction movies, including Jurassic Park. That was dinosaurs, but there's always some idiot that's going to do something stupid with a virus. And if you watch these movies, it's always what? A government scientist, a military scientist, or some evil, James Bond type villain. Oh yeah, I can do this. It's really cool. If you saw the movie Jurassic Park, that's very much on point here. Jeff Goldblum, I love that guy, great actor. Jeff Goldblum, who's the kind of chaos mathematician guy, asked the CEO of the whole Jurassic Park, the English guy, hey, you're like a child who found his father's gun. All you want to do is show off what you can do with it.
Yeah.
I think that's the most accurate statement I've ever seen in a movie. That's exactly what it is. These guys, they just want to show people, look what we can do.
Yeah.
The federal government, Connor, this 20 years ago, they went up to Alaska, dug into the permafrost at some gold miners camp, which was going on up there during the 1918, 1919 influenza pandemic. They pulled these bodies out, recovered the RNA for the influenza, the Spanish flu virus, got that sequence, then went in and made mutations, a regular influenza virus. In other words, they recreated the 1918 strain by going into a laboratory, taking the 8 segments out of the influenza virus, putting them in plasmids, then just doing simple mutagenesis. And they recreated the Spanish flu in a laboratory.
Yeah.
They didn't tell anybody they were doing this. They just did it. Okay. Then they published it. And when I saw that come out, I said, what the heck are you guys doing? That's probably why there's no more Armed Forces Institute of Pathology. But,
Yeah.
I mean, it's easy to do. I actually had one of those kids in my class. One of the technicians was in my class at Hopkins that was involved in that. Nice girl. But I mean, what do you guys, what were you thinking when you did that?
Yeah.
But look, you don't need CRISPR to do this. There's like a dozen different ways you can do the same thing. CRISPR makes it easier and much more exotic, but you don't need that. You can do it other ways.
Yeah.
And just to set up the CRISPR, it's a lot quicker just to do PCR and do it that way, or homologous recombination. And the only time you lose is screening for the virus, which it takes a little bit of time, but if you know what you're doing, it's not so bad. That's why God made graduate students, right? So that's. I mean, go ahead.
Oh yeah, I was just gonna say on the topic of like people kind of asking, doing first and asking questions later, begging for forgiveness, not permission. Do you think that like something like CRISPR needs to be more regulated and like closely looked at?
Should we be controlling this stuff? Sure. You also maybe want to control the people that make oligonucleotides. Everybody watching this probably knows what an oligonucleotide, right? Single stranded DNA.
Yeah.
So if you, these things are all robot now, right? So if you have the sequence of smallpox, okay, and by the way, it's hard to get that now. We've taken it off the air, but if you have the sequence of a virus, you could just make 20 to 30 spare oligonucleotides. And when you make them, so they overlap on each other. Then you throw them all together in a tube, and you heat that up to boiling, and you put it in maybe a 500 or 1,000 milliliter beaker, and you just put it in there, boil it for 5 minutes, and make sure everything's all mixed up. And then you take it off and you let it cool down slowly. Well, just by the laws of thermodynamic, those are all going to line up and find their counterpart, and they're all going to anneal. When you do, you're going to have a gene segment.
Mhm.
So you make each one of those genes separately, then you hook them all up together, you recreate a virus. People have already done this with a couple of phage. Okay, one of them they did it with mycoplasma. Okay, the little short genome sort of a bacteria. Smallpox is only about 200,000 kilodalton, 200,000 base pairs, 200 kilodaltons. Mycoplasma is like 400. So I mean, you've already exceeded your bandwidth there, so you can make that. We have removed some of the genes from smallpox, but off the air, off the internet. But still, I mean, other countries have that, and Russia has smallpox. We have smallpox. So yeah, you got to be careful when you start fooling around with this stuff. As far as controlling the government controlling CRISPR access and that sort of thing, I don't think that would do any good at this point. It's too late. The cat's out of the bag. And we do have programs looking to see if somebody is making, oligonucleotides that would, be consistent with a select agent, like a virus. Viruses are much more easy to recreate than a bacteria. They're much larger genomes. But, I mean, it's kind of child's play for someone like me and I mean, even some undergraduate and graduate students. With the proper instruction, they can go into a lab and do this. So what we worry about is not so much as CRISPR out of control, but who has access to a
Yeah.
laboratory where they can do this? Where's that stuff going to end up, right?
Yeah. I
So, frankly, I think it's too late for the government to control it. Everybody's got it, and there's all kinds of papers out there now how to do it.
yeah.
So, I mean,
I completely agree. I think I guess on the topic of the government kind of controlling CRISPR, while the cat is already out of the bag, in terms of what they can control, do you think that they have like a duty to try to research this as much as possible to
oh,
eliminate
sure,
things
yeah,
like,
yeah. I mean,
like
we're
sickle
looking at that.
cell?
I mean, the government funds a lot of work, and you're looking at people sending in to broad agency announcements, BAAs. There's also grant season where you can submit a grant, an R01 grant or whatever, to the National Institutes of Health or Center for Disease Control or whatever. You can request research money for that. Okay, we're reading those things carefully. If somebody says, "Hey, I'd like to use CRISPR to recreate 1918 endemic, epidemic flu virus," maybe you're not going to get funded for that. Or listen, I want to make a virus even more infectious than COVID-19. Okay, there's always some idiot that's going to do something stupid, like I said, with a virus. But we hope to stop a lot of that in the beginning. You could try to stop it, but you're not going to stop it. So, the cat's out of the bag. So all we can do now is try to surreptitiously watch for that, and hope that if somebody's doing it, we get wind, and we hope for the professionalism of people in this country. But this isn't the only country. America is not the only country where you have people that are trained in biology, and they're not stupid. Some of them are very well trained.
Yeah, I agree with that. So in terms of like germline editing,
Mhm.
should we allow like gene editing in embryos, which could be passed down to future generations and
Yeah, yeah,
like
yeah.
potentially
I was reading
alter
that
permanently?
question on here. Yeah,
Yeah.
that is a that is a very difficult ethical question for someone like me to answer because I know where the pitfalls are. And if you're going to use like CRISPR for that, you know, CRISPR's not perfect. It can insert in other places besides where you have your guide RNA. Okay,
Mhm.
if you look at a germline of a cell, I mean, these experiments have been done, maybe not published, but if you take like green fluorescent protein, all right, and try to CRISPR that into an embryo, I've actually done that with retroviruses. You could take a stem cell, like a hematopoietic stem cell,
Mhm.
and get it at a certain stage where it's differentiated, like into a B cell or a T cell. We can infect that with a retrovirus that carries green fluorescent protein, for the jellyfish. And the results of those things were incredible. You can follow where every one of those daughter cells go in a mouse. Okay. And it's kind of gross the way you do it, but you could actually slice a mouse and make thin sections of a mouse and hit it with UV light and see where all these green cells went.
Hmm.
So I know it's incredible, but it's easy to do. And it allowed us to track and see how a lot of this stuff takes place. Well, you know, if you look at the number of copies that CRISPR can put into a cell, yeah, it's going to go where you want it to go. If you want to add some gene or correct something, okay, but that doesn't mean it's not going to go elsewhere. CRISPR's not perfect, and when you use a lot of CRISPR, a lot of guide RNA and a lot of your inserts, that could go other places in the genome. It could be anywhere from 10 to 100 to 1,000 copies. Depends upon how active the DNA in the cells being processed. So, you know, that is fraught with problems because if you're fooling around with an embryo and it's a human embryo, look out
Yeah.
because you don't know what the heck you're going to get. So the ethical dilemma there is tremendous. You know, I mean, I joke around in class. I used to teach a cell biology class at Hopkins. I joke around and said, look, Star Wars movies, the Clone Wars, right?
Yeah.
That's not fiction. That could be done. If you were crazy enough to do it, you could make 1 million cloned soldiers, right? You get like Rambo and breed him with Ripley from Aliens, right? There's a tough soldier right there, right? And then you get those embryos out, and you go to like the first few replication stages where you got dozens or hundreds of cells, and then you gently proteolytically disrupt that embryo. It hasn't differentiated yet. You can now make you get like 100 cells. You make 100 embryos out of that. Then you can do it again, so it's 100 times 100. Then you got 1,000 times 1,000. You can make millions of soldiers that way, without any fancy CRISPR or anything.
Dang.
Okay, because you were careful how you bred them. And think about it for a second. All the blood type is exactly the same. All the organs are exactly the same genetically because it's all from the same embryo. You can transplant stuff back and forth. You can do all kinds of things with that, and it's tough guys, right? You genetically engineer them. I would not be surprised if somebody right now is not doing that, but it can be done very easily. I think
Huh.
I would never do it, but there's always some nut out there that will.
Yeah, yeah, that's it's actually a very interesting point. Yeah, I guess to kind of move that a bit into the topic of like designer babies, which is one of the more controversial parts about CRISPR.
Yeah,
Due to
yeah.
like
Well, that's the end result of that is Gattaca, right? The
yeah,
movie, which I thought was very accurate, not scientifically, but I mean, what would happen if you actually did that?
yeah, yeah. Do you think like we have a moral right to do stuff like that where because I mean, at the end of the day, in my opinion, I guess, anyway, I think stuff like designer babies will just go to the highest bidder. Do you think that it should be like that, or do you think there should be like steps in place to make it so that everybody can have access to these like stuff and
Yeah, no, I
possibly
see where
work
you're
for
going with
them?
this. Yeah, I think that's the ultimate biological nightmare experience is you start doing this and you do something that you didn't realize you were doing. At this point in development, I don't think we know enough about human genome science and kind of human chromosomal structure to actually do something like that. And my problem is, I was raised up, by Jesuit, priest, right?
Mhm.
So that type of stuff you don't do. But from a scientific standpoint, or just a moral, ethical standpoint, I think you're taking a hell of a risk to take a human being and alter them that way. It's been tried not in utero, but it's been tried in young kids. For example, bubble babies, right, that don't have their immune system intact.
Mhm.
You can insert. There's a couple of enzymes missing in white blood cells. You can take bone marrow out, isolate those cells by marker analysis on a flow cytometer, and insert a virus that carries the correct gene, and that'll go right to the point and insert. Now it's making that enzyme. Then you can take those cells and put them back into the patient. Well, when we first started doing that, I actually participated in this. We first started doing that. They killed some people, not me personally, but they were using adenoviruses to infect cells and put the right gene in.
Mhm.
And one kid up in Philadelphia actually died because he had probably been exposed to adenovirus earlier in his life and had a very, very vigorous anamnestic response, like a very bad allergic response, and then died from a major inflammatory case. And we failed a couple of times doing it. So now when we do it, we take bone marrow cells and try to infect them and then put them back into the patient, once you get all the stuff out. I mean,
Yeah.
we're still in the infancy for that.
Mhm.
And I think we're way far off from being able to do that with CRISPR or anything like that in humans. And also, it's the ethics, the medical ethics in this country. Anybody that was that had that knowledge, that was working in a reputable biomedical research institute or big hospital system or something, that got caught doing that, boy, oh boy, I think you'd be in trouble. And
Yeah.
I think the ethics. Forget about the scientific. Could it be done? I think eventually it could be done.
Yeah.
But the ethics of that is wow. Okay, what if you screw up and this kid comes out, leg growing out of his head or something because
Yeah.
you did something and interrupted normal gene development. I took a couple courses in developmental biology. Was always interested in this, and Hopkins has a nice course in developmental biology. Uh, yeah, the undergraduate school. We just don't know enough. I would never ever do an experiment like that because of what could happen to the offspring,
Yeah.
and not to mention that. I mean, what happens if the kid comes out severely retarded or very very physically disabled or physically malformed?
Mhm.
You're responsible for that.
Yeah.
So I just don't think it's safe at this point to do something like that.
Yeah, I
I
completely
think it's
agree.
I think it's an actually dangerous experiment, but that doesn't say some idiot's not going to do it,
Yeah. And I guess
or hasn't done it already.
Yeah, and I guess on the topic of that, informed consent, super important, if the embryos or future generations, are effective, who gives
Yeah.
consent and like how do
Well,
we
there's informed.
consent?
Okay, there's your by definition. There's your most difficult question: is informed consent. I mean, you're a biology major.
Yeah.
I'm sort of a biology major too, but if you weren't a biology major, you were just like a truck driver or a bus driver or something, okay? And you come to me and I'm, MD, PhD in charge of the, OBGYN department at ASU or something. And you come in, oh, Dr. Hodge, I'd like you to correct something in my baby, okay? And like for an insane second, I said, yeah, okay, I'll do it for you. Do you really understand as a truck driver or a layman what the hell you're talking about and what could go wrong? And I'm telling you, well, the kid come out, arm growing out of his chest, or he could come out looking like an alien, or who the hell knows what could happen.
Yeah.
Do you really want to take that risk? Oh yeah, I want my baby. Okay, so this is where informed consent breaks down. Uh, you can always think you're giving a person informed consent. Look, we have this chemotherapeutic drug we're going to give your dad. Uh, he wants to take it. We're just checking with you because you're his legal representative, whatnot, right? The power of medical attorney,
Mhm.
medical power of attorney. And we want you to think about this for a minute and give us your informed consent. Well, you're not an oncologist. You don't know how this stuff works. He's saying, well, there's about a 1 in 3 chance that it could kill your father or make the cancer even worse. Do you want to take that risk? Oh, sure. Yeah, right. Because a doctor's sitting there telling you white lab coat, nice tie,
Yeah,
expensive shoes. You're going to agree. Okay. That's not informed consent. That's just kind of, I don't want to use the word browbeating, but you think you're getting informed consent and they've signed a form, but that's not informed consent.
yeah, not
So
the whole
as
story.
medical ethicists and doctors, we're supposed to know better than to ask a patient a question like that. Now, that's one thing. Okay. You got somebody that's sitting on death's doorstep and they're like in stage 4 cancer, and you have an experimental drug that might work or might kill them. Look, I could give you this. It might kill you, but it could save you. You're going to be dead in a month. What do you want to do? What would you do? You'd take the drug, right?
Yeah, I would. Yeah,
Okay. I would too. Yeah. So that is what informed consent is. It's not like, oh yeah, we think we can fix your baby. No, 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 no. Don't do that.
yeah,
I think that is, to use a military term, a bridge too far. And I think that you're way off the ethical reservation. You're way, way, way outside your comfort zone. But like I said, there's always some idiot that will try it.
yeah. And I think
Maybe
that,
not here, but there's always some idiot out there.
yeah, and I think that's kind of the one of the biggest issues I've seen when I've been researching CRISPR is like it's the way it's marketed is kind of like oh, this will fix it. It's not like it doesn't really dive into
No,
actually like
no,
what it does. It just kind of
yeah.
says this will
If
fix
did you
everything.
read you read about CRISPR? You know how it works, right?
Yeah,
I mean, it's not that difficult to set it up. And the guys that did that, I think, got the Nobel Prize that found that. But you know, that is not for humans. That's in bacteria. So I mean, it's a little different thing. But yeah, I think that people that read that get the wrong idea and they think they can do anything with it,
yeah,
or that they can do it and not get in trouble.
yeah,
So it's the rule of what the hell, you know, no good deed goes unpunished, right?
yeah,
You want to try to help people, but you end up making a matter worse. So I mean, I hate to use the word "let God decide what happens," but ultimately, you got to think about what's the consequences if I do this and it doesn't go right.
yeah,
If it goes right, you're on the cover of Time magazine,
yeah.
woohoo,
Persevere.
and you
Yeah,
get a Nobel Prize. If it goes wrong, you're like Dr. Mengele. Yeah, and believe me, in my travels over all these years, I've seen people, I've worked with people that I think would be capable of trying something like that and
yeah,
not even blink, not even blink. They would, yeah, science has to go forward.
yeah,
Like, let's keep funding COVID research, or let's keep funding, a gain of function research
yeah.
until it jumps up and bites us in the ass. Then it's not such a cool experiment anymore.
Yeah, and with the viewpoint of like, science needs to go forward, like no matter
Yeah,
what, do you think like government should regulate CRISPR's use like for bacteria to prevent like potential like superbugs or
yeah.
antibiotics?
Well, we're certainly looking at that, but we're looking at it from the standpoint of a regulatory. I mean, if somebody's going to do it, they're going to do it. You can't stop them.
Yeah,
Okay, they're going to do the experiment on the side. But for trying to get grant money to do that, we could stop it by not giving them grant money. And then there's congressional laws right now being written about this. But if you did something like that, like in a major institution, you'd be cooked. You'd never work in this town again.
yeah.
Okay, you're out, and nobody would pick you up. Not in the United States.
And with all that, like people just like moving and all that, do you think transparency is like a really important thing? Because I personally think, or I'm of the opinion that like any scientific research should be like completely transparent to the public.
Sure,
Like
yeah,
something like this should be like, kind of like as transparent as possible with every step of the way with research.
sure, yeah. And you have to. A lot of these things, anything that's going to the federal government, they're going to look at it. And there are very strict rules and ethics. I mean, you have to look at this, and they have, like, study group supervisors and people that look at all this. You have to have institutional permission to do a lot of these things. And if it's something controversial like gain of function, God forbid, or if you're doing something with human embryos, look out. They're going to be looking at you, pardon the pun, with a microscope, okay, and making sure you're not going to do that. But again, that doesn't stop some idiot from running out and doing this. We can like, control it by withholding funding, okay. But what if you go to like Al Qaeda or one of these groups, Taliban nuts or something? Look, after the 9/11 attacks, those nuts are over there looking to make anthrax. We know because we were looking for them. Okay, they were trying it. Okay, and they tried several different attempts, and unfortunately, having a 2,000-pound bomb fall on your head kind of screws up your laboratory.
Yeah.
But they were doing it. They were trying it, and not only that, the scary part is the ones we don't know about.
Yeah.
Those are the ones that are. It's Donald Rumsfeld wrote it in a book. It's like it's there are known knowns, known unknowns, and unknown unknowns. So the unknown unknown, that's the one you got to worry about. You don't
Yeah.
even know they're doing it. You don't know who's doing it, but you know some idiot's trying it.
Yeah.
Now they may not be successful. We hope that as laboratory scientists and ethical scientists, people won't do that. Well, look, not everybody is ethical, and don't sell these lunatics short because there's always some nut to go out and do something like that. My
Yeah.
opinion. And I
And
mean, we had major discussions for years about this. We used to do the biological threat and risk assessment. Okay, BITRA. I mean, we did that all the time. What are the chances of this happening? And we had hundreds of scientists looking. We had a questionnaire, big long questionnaire. All that stuff got put into a computer, and we kind of, racked and stacked all that and figured out what's the chances of this happening, what's the chances of that happening. I mean, what's the chance that somebody can go in and make smallpox?
yeah.
Good. Okay. I mean, it's easy. I could do it. I mean, if I had the sequence, I could do it. But would I do it? No, of course not. Why the hell would you want to do that?
Yeah.
Yeah. But look, I mean, like I said, somebody. But then again, the people that we worry the most about that could distribute something, you can make the agent, but you have to figure out how to distribute it, how to get it into your target population. So, I mean, that's acquisition of agents only step one. Then you got to figure out how you're going to grow it, how you're going to distribute it, so on and so forth. How you're going to conduct an attack. So, this is not biology. This is biowarfare.
Yeah.
So, I mean, you hit somebody, you can vaccinate people, but you hit somebody with 1,000 or 10,000, infectious doses. Vaccine's not going to protect you.
Yeah. So I think I only had basically one more question, and
Okay.
my question would be, do you think the pros of CRISPR basically outweigh the cons? The pros of eradicating genetic disorders and stuff like that versus the cons of biowarfare. Do you think it's worth it, or do you think this is something that you know?
Did you see in the papers and the media the other day that these guys tried to bring a wolf species back called dire wolves?
Oh yeah, I saw
And
that.
they
Yeah.
went in and made like maybe a dozen mutations and wolf embryo and then bred them. Um, I think that's a very simplistic view of like you just all of a sudden you just make these things and now you're making a dire wolf. I think it's a lot more complex than that. This isn't Jurassic Park, okay?
Yeah.
You can't just fill in the rest of the spaces with reptile DNA. It doesn't work that way. I mean, you're dealing with millions, sometimes billions of base pairs, and everything has to be perfectly positioned. So, with respect to your question, do I think it's worth it? I think that the ability to correct genetic mutations in people, you presuppose that you already know the mutations there. Like, for example, somebody, Hebrews, they carry like a couple of genetic diseases because there's a lot of closed breeding there between the different groups and that religion. So, for example, Tay-Sachs disease is a very common disease among Jews. If you knew you had it, could you correct it? Maybe, yeah. I mean, but how the heck would you do that? Okay, you have to get the individual sperm and correct it, or the individual egg and correct that. Could you breed those things together in vitro and then go in and do the correction? Yes, I think you could. But then it brings up a question: is it really is it ethical for one, and is it safe? I just don't think we're at a position now, technology wise, where we can do that and just go ahead and implant an embryo with a correction in it using CRISPR and hope everything is going to work out for the best. CRISPR doesn't. I mean, you hope it goes where you're aiming it with your guide RNA and everything. You hope all that works out. But looking at this thing, historically, CRISPR can insert non-preferentially in other places as well. In other words, it's not completely as specific as we thought it was. Yes, you can do a correction, but where else is it going to go? And what genes is it interrupting? So you go through, 40 weeks of gestation, and then you have a baby. It was an embryo, now it's a baby, and it comes out, and you may not know you did something wrong for several years. Or maybe the baby is stillborn. Maybe something else goes wrong. We still don't fully understand the detection and the corrections of what goes on in utero. Some kids don't make it all the way through. They're miscarried or spontaneously aborted. What causes that, we don't know. Okay, so I think you're in an area where we're not quite ready yet, and so that relegates us to looking at animal models first. Well, whatever animal model you're looking at, you're going to have to get into probably non-human primates. You don't want to do it in an inferior species like a mouse or a cat or a dog or something. You want to get up into primates, okay? Then the question is, are you getting into some neurological functions or brain functions, right? CNS stuff. And then if something comes out in a non-human primate, an ape, how do you know? So, I mean, they live a long time, a lot of them, 20, 30, 40 years. But even that has moral implications. I mean, some people don't want you experimenting on animals. So the misuse of CRISPR in kind of biowarfare and creating new organisms, in a lot of cases, you don't even have to use CRISPR. Okay, some of these things will kind of self-select and give you some pretty bad outcomes biologically. So the question is, it's an interesting question, deep question. Morally, what do we do? And if you're asking me personally, I'm not quite ready. If I was sitting in the front office and that grant proposal came across my desk, I would say, no, I don't think so. It's too dangerous yet for humans. And I don't know how long that's going to last, but I think some countries that don't have any qualms about doing that, I think they would go ahead and try it. I think it's dangerous. And biology, if you're doing this with insects or some people suggested, well, let's just vaccinate people using the same mosquitoes that are giving them whatever, okay? Well, you realize how dangerous that is? If you start getting into the genome of an animal, and especially an animal that you're not going to release in nature, and that thing starts breeding, I mean, what did you create? So, I mean, you could select for something. Maybe you did something where CRISPR inserted into the wrong gene somewhere, and now you make the thing resistant to insecticides or resistant to whatever. And so it's a dark, nightmarish scenario if that ever gets out of control. How do you stop it? So my personal feeling is, and I think a lot of governments would agree with me, governments with common sense, that's too dangerous right now to do. I don't think we should be doing it. I think there is a danger with CRISPR, but I mean, look, that danger's always been there. We didn't even have CRISPR. Look at all the bioagents we've dealt with over the years, and
Yeah.
nature's done it for us. Nature made highly pathogenic influenza, and then you have stuff that you can get right outside of Flagstaff. You can get, bubonic plague and hantavirus and everything else. It's just out in nature. So I think if you're good enough to go into a lab and use CRISPR, that maybe you don't even want to waste your time. If you know how to
Yeah.
recover these agents out of nature, and I'm one of those guys that do, I mean, it's just go get what nature already prepared for you.
Yeah, no, I think that is a great response. I actually had one more from my class. We were just talking about this in my capstone class,
Yeah.
and it got to kind of a heated debate. So I was just kind of wondering your 2 cents on it since it's fresh on my mind. We were debating kind of like where to draw the line in terms of that because like now you can do like I guess there you can start doing like picking stuff as like, trivial as like the eye color or like how like defined your nose or whatever it is.
Yeah. Yeah.
Do you think there should be a line stopped at like just genetic disabilities, or do you think we should do, you know, just do whatever, it's your,
Well, there's two answers to that, as always in these cases. The first answer
Mhm.
is the kind of ethical, moralistic question, and that is, do we want parents to do that type of stuff? Now, you can't
Yeah,
do
not
egg and
frowned
sperm
upon.
separate. You got to do an embryo. Okay, so you're already relegated down to, in vitro fertilization.
Mhm.
And then here's the question: do you want to take a risk with your offspring by doing some kind of genetic manipulation of the embryos? I think it's actually dangerous. But that look, this is the other part of the question. That doesn't mean people are not going to try to do it. I can foresee in the next few years offshore. Okay, hey, you want to do this? You want to do that? Just come over here. You don't have to do it in the United States. I could see a vacation, like a genetic vacation for people that want to change the color of their baby's eyes or want to do this or want to do that. Now, a lot of stuff we don't know. I mean, we know where those genes are at that do pigmentation, but I mean, do we really want to allow that type of stuff over here? I don't think you're going to see that. I think not even on a moral basis, on a scientific basis, I think it's too dangerous. We just don't know at this point, which means that opens it up for these private companies that do that type of stuff to figure out how to do it. And then the moral, ethical part is who are they going to do it on?
Yeah.
There's plenty of people out there that will donate eggs and sperm for money. And these are multinational corporations. I mean, I could see big pharma surreptitiously farming something like this out and see if they can do it. Uh, Look, it's Brave New World. 50 years later, and even more in some of those books, they had no idea what our capabilities were going to be genetically. Okay,
Yeah.
so
So you
you
think
see
from
the
like
you see the danger there, right?
the US, yeah, yeah, 100%. So
Yeah.
just you think from like the US government standpoint, like there should be a line, but you think like people will just do what they want to do.
Well,
That's kind of that's
yeah
kind of where my
yeah
opinion is.
we can't control private enterprise. We can't stop people that are not
Mhm.
trying to get federal funding. Okay, that we don't even know what they're doing. They could say, "I want to make I don't know the hell, yeah, tomatoes that are resistant to freezing." Okay, or strawberries that are resistant to freezing, right? And so I want to make
Mhm.
the antifreeze gene in flounder. Flounder has an antifreeze gene, so they don't kind of go like that in their frozen fish because the North Atlantic
Yeah.
gets pretty cold. So I had a guy when I was at Hopkins, that was his dissertation project. He got the flounder gene and extracted it and PCR'd that thing out and put it into strawberries and tomatoes. But he was
Nice.
really heavy into crop genetics and all. And
Mhm.
this same guy went on later on. to make corn and other, genetically engineered, what they call them, GMO, genetically modified organisms. And
Mhm.
then he was able to, you know, make these things and sell them. He's a very wealthy guy now. I mean, you always have,
I
yeah.
imagine.
So, and it was a simple project. He just took one gene from one organism, put it in another. Now that was 40 years ago, in the mid to late 80s. But it can be done. And I mean, the question is, do you want to do it? Uh,
Mhm.
so, as far as catching people doing that, that would be caught probably if they went for a federal grant. These guys that read the grants are given very specific, requirements for a grant. And if they're doing
Mhm.
something like that, making a GMO, they're going to look at that closely. So if you say, "Hey, I want to make this or I want to make that," they're going to look at it pretty closely, just because of what the guy's trying to do and the girl's trying to do. So I
Yeah.
don't know. I don't know where all that shakes out, but you just have to be very, very careful when you start looking at these things and say what's the end result of what this person is trying to do. Now, if somebody wants to do gain of function work in a virus right now, I don't think that's going to be approved, especially under the current administration. But you never know.
Yeah. Yeah.
So, I mean, we're opening the door here to what could easily be called the brave new world. And boy, oh boy, it's there's some danger in there. And I mean, CRISPR has been oversold. I think you can do certain things with it, but I just
Mhm.
don't think it's ready for prime time. There's certain things you can do with it, certain things you can't do, and it's not perfect, like I said. So I mean, it's a little dangerous to assume all this stuff is just going to be easy to do. Plus, you got to know what the heck you're doing. The average
Yeah.
person couldn't do that. I don't think it's beyond the possibility, realm of possible experiments where you could take an entire human being and isolate all those chromosomes and sequence the chromosomes and then look and see where are there any mistakes in that human being. So the movie Gattaca, okay, which is highly speculative science fiction, when it was written,
Mhm.
that's not so highly speculative anymore. I don't want to say anything's possible because it takes money and time and effort, but
Yeah.
anyone that puts their mind to it and get into this, get trained up in it, and be pretty dangerous.
Definitely agree with that. Yeah.
Yeah,
Well, I think that's all the questions I have. I want to thank you like very much
yeah, no
for
problem.
taking the time out of your day to talk to me.
See you later.
Thank you guys for tuning into Editing Humanity. Be sure to check out our other videos and follow us on social media, and I'll see you next time.